All right, hello everyone, and welcome back to Bio 420. So here we're talking much more about respiratory viruses. So what are respiratory viruses? As their name says, these are a group of viruses that usually can infect epithelial cells that are found both in your upper, so around this area, and your lower respiratory tract. So of course, this leads to different, a range of different, let's say, side effects. If you have much more, let's say, up in your upper respiratory tract, you get your, let's say, common cold symptoms. This could be your runny nose, your bit of a sore throat, and coughing. But of course, if they start to go specifically to your lower respiratory tract and specific your lungs, you're going to get a lot of issues with pneumonia and acute respiratory distress. These could be extremely fatal. So respiratory viruses, why we call them like this? Because we call them professional respiratory viruses because they're able to really specifically replicate within our respiratory system and induce a lot of clinical manifestations around there. Because it's not that if a virus enters through our respiratory tract that it means they're going to basically induce respiratory symptoms. That's not true. You have measles and varicella. These are two other viruses that are able to pass in through your respiratory tract, but they usually do not, let's say, colonize those areas, but they actually go more systemically. So that's why we group them together within the respiratory viruses. So these are coming from many different types of viruses. These could be DNA viruses, RNA viruses, enveloped, non-enveloped. So there are different families. So per se, for that stuff, one thing that's unifying among them is their ability to really infect the mucosa and the surface areas. But also one other thing is these viruses are highly transmissible, and they usually are, kind of infected, like kind of their transmission is through respiratory droplets, like aerosols or direct contact with contaminated surfaces. And because of their high infectivity and their transmission, they actually are the biggest responsible viruses when it comes to global morbidity and mortality. And this is specific with people that are immunocompromised or in generally do not have a good immune system, like people like your infants or elderly people. So they're a very important group of viruses to study. So the general properties of viruses, as I said, these viruses can be different types. They could be DNA and RNA. But the majority, the clear overwhelming majority of respiratory viruses are RNA. And because they are RNA, you have their own RNA-dependent RNA polymerase. They need it to replicate. But the issue with this polymerase, it has a lack of proofreading. So because it has a lack of proofreading, you get a lot of mutations. So what happens is you have a high antigenic or high antigenicity. So these viruses keep evolving, and more and more and more. They have different types of, let's say, different viruses that can come up. And because of this antigenic variation, you have immune evasion and you recurrent infections throughout life. This is why we get infected. We have common colds constantly. Why we get infected by influenza more than once. You name it, because these viruses keep literally mutating. So the majority of them are RNA, but you have an exception. This is adenoviruses, and adenoviruses are DNA. So they're made of DNA viruses. They do not actually have this huge antigenetic variation per se, but in generally they cause a significant respiratory diseases still. They're quite powerful. This is purely because adenoviruses, they are, enveloped. So they're non-enveloped. Sorry, my bad. They're non-enveloped. So they only have a capsid. And as we know, having just a capsid actually makes you much more resistant towards the temperature and basically the outside environment. So other respiratory viruses, the majority of them are enveloped, so meaning that they have a lipid membrane derived from the host cell. So these ones, they still are, of course, in some sort of way stable, but they're not as stable as your your capsid, your non-enveloped viruses like adeno. And again, there be many families that can be under the respiratory viruses. This could be your orthomyxoviruses. These are, for example, the family of viruses that include influenza, paramyxoviruses, which are including parainfluenza, RSV, and the human metapneumovirus, coronaviruses, picornaviruses like rhino, and adenoviruses. So again, many different types of families that are basically much more, you differentiate them between if they're DNA and if they're enveloped or not enveloped. So despite them being from different families, they're able to infect and replicate in respiratory epithelial cells. Again, there are professional respiratory viruses. So their inflammation really causes a lot of mucus production. It causes a lot of damage to our respiratory lining. So this is all of these things causes our clinical symptoms like the coughing, the congestion, the difficulty in breathing that we get accompanied with these respiratory viruses per se. So sometimes understanding these viruses are very important because they have a unique mechanism of replication. Based on this, we can actually devise novel antiviral therapies. Basically, every single one of these have a different immune evasion and pathogenesis. So we can basically sometimes use certain clinical features to distinguish among these viruses. So when it comes to diagnosis, generally these viruses, other of course, some of them have specific symptoms. Majority of them have very common symptoms, which is the cold-like symptoms. So in generally, by going to do a clinical basically kind of visit, the doctor's not able to diagnose you by looking at your symptoms. It requires basically either you do a PCR test. So what you do is basically you take specimens coming from your nasal swabs, your throat swabs, or your sputum samples, and then you look at PCR. So through PCR, you can look at DNA or RNA coming from any of the viruses that you might be infected with. Of course, nowadays now we have these antigen or antibody tests. So these basically antigen, what they do is they look at certain antigens, for example, certain glycoproteins found in influenza viruses or RSV or basically adeno, or you have the antibody test if you have any antibodies against these viruses. So these are your rapid, basically kind of tests that we basically know quite well nowadays. So I want to really dive into influenza today, and I will be talking quite a lot about it because it's extremely important. It's one of the most important respiratory pathogens because it's able to induce two things. Seasonal outbreaks, so we have always a seasonal outbreak of influenza, so influenza that are literally kind of infecting our population every year, and every year the seasonal outbreaks, the influenza is different because of certain mutations, because again, influenza viruses, they mutate quite a lot. But we also have global pandemics. These are a bit more rare. These only happen when you have a novel strain coming up. And these novel strains of influenza, they usually are so new that our immune system is not able to detect it at all. So because it's not able to detect it at all, this virus is able to evade your immune system quite efficiently and do a quite extensive damage. And an example of this is the 1918 influenza pandemic, also called as the Spanish flu. This kind of influenza literally wiped out almost all of Europe. So it actually caused more death than multiple major wars combined together. So there's a massive importance when it comes to clinically, when it comes to influenza viruses. So influenza viruses, there are three types: influenza A, B, and C. And the difference between them, again, is just their antigenicity. So they have different, basically, let's say, antigens within their glycoproteins. So you see if you influenza A, influenza B is literally very similar structurally. Differences are, these differences in epitopes they have within their glycoproteins. And again, here goes with influenza C, differences, just in their ability glycoproteins on the outside. But in generally, because of these differences that they have in their glycoproteins, they are able to, infect different types of hosts. Influenza A is the most important because it's able to actually induce pandemics, because influenza A is able to infect both humans, but also other animals. And this ability for influenza A to bind to both humans and animals gives it its ability to induce pandemics. Then you have influenza B, also causes outbreaks, but it does not lead to pandemics. Usually you have your seasonal outbreaks of influenza B. Influenza C usually causes very mild respiratory illnesses. It's not really associated with major outbreaks. It's not such an important virus. You have usually nowadays your influenza vaccines are trying to deal with influenza A and B. So because C is not that important per se. So how's the structure of the influenza virus? Well, actually it's an envelope virus, so it has basically this host cell membrane that it steals. It actually has RNA, and its RNA is single-stranded, it's negative sense, and it's basically segmented, meaning that it's not circular. So you have these nice segments, and in specific you have 8 segments of RNA. And this segment structure is extremely important because they have segmented portions, they are able, so if you have two different influenza viruses, they're able to exchange RNA segments so much more easy because they can take, for example, exchange this RNA segment for another RNA segment. And this is what you get with having literally these pandemics, where you have these viruses that are exchanging whole segments. So now you're creating a whole different virus. It's not just a couple of mutations in an RNA that you're changing a beta-glycoprotein. No, now you have a whole different proteins because you took an RNA segment that from a different virus. So this can be extremely, this structure is extremely important because you allow this genetic reassortment, which is technically this passing on of this big RNA segments. The virus also contains helical nucleocapsid, so it's quite its capsid is quite helical, and also you have an RNA-dependent RNA polymerase because it has a negative sense RNA genome, so it requires a RNA-dependent RNA polymerase. So it's quite important in this kind of sense. So when it comes to actually the binding, so as we know, the first, let's say, step when it comes to viral entry is basically the virus have to bind to the host receptor. And usually this is usually done by two major glycoproteins, the hemagglutinin, which we call HA, and the neuraminidase, which is called, we call this as NA. So hemagglutinin is responsible for binding to the host receptors, specifically sialic acids. So sialic acids is gonna basically bind towards your hemagglutinin, and this is gonna induce literally the fusion or the endocytosis of your whole influenza viruses. So this induces endocytosis and will bring it in. Neuraminidase is much more important when it's trying to exit, because when the virus is trying to exit, so it doesn't get stuck to the sialic acids, neuraminidase removes the sialic acids and allows basically the virus to be gonna exit nicely. So both are acting at literally the polar opposite directions. One is in the entry to bind, the other one, so hemagglutinin is binding sialic acid to enter, neuraminidase is removing sialic acid, so basically it's allowed to kind of exit. So it's super important in order to do that in per se. And then generally, there's been now new types of drugs that are targeting neuraminidase in some sort of way. But of course, they're vaccines, so we know that influenza vaccines, they're basically built to design antibodies able to bind to both neuraminidase and your hemagglutinin. So you're able to stop this exit or entry of the virus. So now comes your antigenic drift and shift, because as we know, the RNA polymerase it doesn't have this kind of proofreading mechanism. So what happens is that you do have mutations accumulating, specifically in your neuraminidase or your hemagglutinin. So basically you have, these small little differences that can actually evade antibody responses. We call this antigenetic drift. And this drift, means small gradual mutations, either in the hemagglutinin or neuraminidase, and these are the ones responsible for influenza outbreaks. Every year, the influenza basically gains these mutations. So the rate of mutations is quite high that it accumulates. So that's why every year we need a new vaccine, because basically when you create a vaccine against, for example, this virus per se, that has this kind of, let's say, oval glycoproteins, the antibodies will be able to bind. But of course, this virus is going to go through some sort of drift where you have small mutations occurring, and then basically you're going to have a different type of glycoproteins where the antibodies now are not able to bind here. So you need to get a new vaccine. Then when it comes to antigenic shift, no, it's literally where you have reassortment of RNA segments. So you have the virus, basically the whole RNA segment, being exchanged with another RNA segment from another virus. So you have these three different viruses, and they're literally kind of shuffling or reassorting their RNA segments. So you create a virus that has completely different hemagglutinin and neuraminidase, which now is completely new, and basically our immunity has a lack of protection against it, and this can be extremely dangerous. And this is what the antigenic shift is what causes, per se, in general, these pandemics. And one important thing that we see in pandemics is the role of animal reservoirs. Because, of course, as I said, specifically influenza A is the main one that causes pandemics. The reason why it's able to cause pandemics is because it's able to bind or kind of infect many different species. These could be marine species, bats, aquatic wild birds, domestic poultry, pigs, and domestic animals. So what you see is you have all these different viruses, influenza viruses, all around, and you have this pig, where we call it the mixing vessel. Literally, this pig is able to be infected from domestic poultry, so avian viruses, so this avian influenza viruses, and also the human. So inside this pig, you have literally a mix of different influenza viruses, and there is where they're able to actually do the reassortment, because now you have different viruses that are able to exchange their RNA segments, and you create novel viruses that are then able to literally kind of infect us and other poultry. So this can cause pandemics. So as we said, usually when it comes to your attachment, we know that usually influenza binds through its hemagglutinin to sialic acids, and this really what it does, it induces in some sort of way your endosome formation. So it causes endocytosis. This basically virus now gets inside a endosome. Within the endosome, what occurs technically here, as we know, the influenza releases this M2 basically kind of ion channel. It has this ion channel that literally puts in the pore of the endosome, so in the membrane of the endosome, and it allows basically hydrogen protons to get inside. So when hydrogen protons get inside, pH decreases. This pH decrease basically causes a whole configuration which allows the influenza virus to uncoat and release literally its basically its RNA, so its genome. So the genome is then going to go and it's going to infect the nucleus. This might seem counterproductive because remember guys, RNA viruses usually they replicate inside the cytoplasm. But influenza virus is one of those kind of viruses that doesn't do that. It has it's one of the exception ones. Similar to what we see with polio. DNA viruses but really replicates in the cytoplasm. While influenza virus is an RNA virus that replicates in the basically nucleus. And this is purely because this RNA virus actually has segmented RNA. So segmented RNA is much, in some sort of way, very, let's say, it's not that stable. It's very reactive and it's able to break down quite a lot. So what it does is that when the RNA from the influenza goes inside the nucleus, it's able to snatch something called the cap, the 5' cap. This is a cap that we have in our basically mRNA. And this cap, what it does, it makes your mRNA much more stable. So what the viruses do is they snatch this cap and they put it in their own viral mRNA. So now this mRNA is able to leave and is much more stable and it increases really its translation. So by snatching this, you do two things. One, you stop the host from replicating its mRNA, so not replicating, so translating its mRNA, but also you help the virus now translate its basically proteins. So this is why influenza really needs to go into the nucleus because the cap is found there. It needs the cap, so that's why it goes to the nucleus. So once basically, it snatched the cap, it starts to translate its proteins. Of course, a lot of the proteins will be done outside because we have the ribosomes and all the basically translation machinery is outside in cytoplasm, so they will be created outside. Some of the viral proteins will go back into the nucleus to further help in the replication of the RNA genome, but some of it will stay outside. So what we see is that the new synthesized viral RNA segments will associate with nuclear proteins and matrix proteins, so to form ribonuclear complexes. So these ribonuclear protein complexes will be able to be transported away from the nucleus, then go into the membrane. In the membrane, you already have your hemagglutinin and neuraminidase, literally, and they're literally waiting for this ribonuclear protein complex to come in. And then, of course, your RNA segments, and voila, you actually have the formation of your capsid, everything, and it literally buds off. So one important thing is that neuraminidase, again, here plays a super important role because because there's a lot of sialic acids that are binding here, keeping this basically from literally budding off. So neuraminidase removes the sialic acids. If neuraminidase is not allowed to do its job, this will stay here and will not bud off. So it's extremely important. So when it comes to transmission and the epidemiology of influenza, as we know with a lot of basically these respiratory viruses, they go through respiratory droplets. So we have these droplets that are come basically through coughing, sneezing, and talking, and they're able to kind of infect almost about a 2 meter distance. So that's why during COVID, they used to say, please have a space of 2 meters or not, because these aerosols have a literally distance of 2 meters. So that's usually how this is transmitted, and why influenza is extremely infectious is because of these aerosols. So we don't need contact. It's just being within our vicinity. And if we're sneezing or we're talking or we're coughing, we produce these aerosols that are able to infect us. So then basically, usually the seasonal influenza typically occurs in the winter months. Usually it peaks in December to February in the northern hemisphere, and then it rapidly spreads throughout crowded environments like schools or healthcares technically. So usually we have annual epidemics because, again, every year this virus, the hemagglutinin or neuraminidase, is completely changing and having these mutations. So this this antigenetic shifts, drifts, the ones that I was just talking about over here, this drift. So usually influenza infection is primarily localized to your respiratory tract. It's either basically found in your upper or your lower airways. If it's in your basically upper airways, it's not a huge issue because, again, it's just affecting just around your basically kind of your trachea and your basically kind of more throat areas. So you have symptoms that are resembling really much a cold. You basically have, in some sort of way, coughing, mucus production, basically kind of sore throats. But the issue is that if it's able to go down, and specifically what it really helps is neuraminidase. Neuraminidase, what it does, it really will chemically destroy your mucus barrier. So it's going to allow the virus to really go inside to your cells because it's able to remove the mucus quite a lot. And if it is able to go down into your lungs, it can do so much damage. But usually it's not that kills you. It's because you've removed your mucus, you removed so many of these basically cells, epithelial cells that have the cilia, so all these protections, you are at much higher rate of infection from bacteria, specifically Staphylococcus aureus and Streptococcus pneumoniae. So in generally, if an influenza virus kills you, it's mostly going to do it through these secondary infections. So Staphylococcus and Streptococcus pneumoniae, they're the ones that usually do that. So in generally, influenza doesn't go to your blood. It mostly starts in local tissue damage, and that's what it mostly does. It stays in the lower and the upper respiratory tracts. So when it comes to immune response to influenza, one of the most important things is antibodies, because antibodies are extremely good at stopping them from disseminating into other cells. We see that in generally IgA antibodies are extremely important because IgA antibodies are mostly found in the respiratory tracts, so it's much more effective there. IgG can be produced, but it's not that effective compared to IgA. We also have very important cytotoxic T cells, so CD8 T cells, that are super important for clearing infected cells as well. But the issue with influenza, it has an NS1 protein that really stops interferon. So when you stop interferon, you really do stop basically your B and T cells from being activated per se. So this is what we have, why sometimes influenza can be quite successful in causing disease, because it's able to really, let's say, suppress our innate immune system quite well. So of course, as we know, clinical features of influenza, usually the incubation time is about 24 to 48 hours, so in general it's quite quick. And you have, your typical symptoms: fever, chills, severe muscle aches, headaches, sore throat, and a dry cough. But in generally, the presence basically of, sometimes what happens is if the virus keeps persisting, it can go inside and cause pneumonia, especially in your lungs. And more dangerously, it can cause secondary bacterial pneumonia. In some rare cases where you see the virus is going systemically, very rare, it can cause Rye syndrome, which basically is literally infections. This is usually in children when it comes to, per se, the liver and basically the brain. So in generally, diagnosis is usually done with PCR to detect the viral DNA, but nowadays you have so many really good serological tests, these rapid antigen tests that tells us, do we have antibodies against influenza per se. And usually we use neuraminidase inhibitors like oseltamivir and zanamivir to basically stop influenza, because again, if you inhibit neuraminidase, the influenza virus is not able to cleave sialic acids, hence the virus is not able to basically bud off. So these drugs are very effective when administered within 48 hours of symptoms onset. So it's very important that you directly take it per se. But in generally, we rely on vaccination, because if we have a good vaccine that is able to match the circulating strain, so if we have basically a good vaccine that we predicted to have exactly the hemagglutinin and neuraminidase with the certain mutations of this pandemic, of this basically outbreak, we are able to really stop quite a lot influenza viral infections. But now I want to talk to you about these two very important viruses. So you have basically these avian influenza, which is H5N1, H7N9. So these are influenza A viruses. They usually, basically, as their name says, are more infecting avian species, specifically, chickens. And usually they're very pathogenic if they're able to jump from the chicken to the human. So they usually happens through, in some sort of way, have certain mutations. So they go certain genetic reassortments or mutations. So this could be either you have a whole reassortment or you have certain mutations, so antigenic shift or drift, that is causing now for some reason this H5N1 or H7N91 viruses to be able to jump from these basically kind of chickens into the human, and they become super pathogenic. They can really kill us because we are not usually infected with H5N1 or H7N9. So it's very important that we always check with chicken or other poultry the viruses and make sure that they're not mutating and gaining mutations that will make us jump into a human. So why one other thing, of course, then other than being in general, this virus is able to actually infect really well or cause a lot of lethality in humans. In general, basically the avian viruses like H5N1, it usually binds to sialic acid, but it binds to this 2,3 specificity, or let's say conformation. So the sialic acids are in this 2,3 conformation with galactose. Usually in chickens it's mostly 2,3. In humans we have 2,6 configuration in the upper respiratory tract, but in the lower respiratory tract, it's much more this 2,3. So this virus per se, so in generally, normal viruses in humans, they only stay up in the upper respiratory tract because they're only able to bind to the 2,6. The 2,3, they're not able to really bind in that well, so that's why they're not able to go down. But when it comes to the basically avian influenza, it's able to bind to these 2,3 specificity or more configuration, which are much more lower. So this virus, rather than going to our upper respiratory tract, it goes to our lower. This causes pneumonia and acute respiratory distress syndromes, because what's happening is we're creating a cytokine storm, and where our immune system is so exaggerated, because again, this avian virus is so new that is really causing so much damage to our lung. Another thing that we also see that's also super important is this H1N1. This is for a swine influenza. So what we saw, specifically this was a pandemic in 2009, we saw that in pigs, these pigs were literally having this H1N1, which is the swine influenza, but it basically was going through some sort of basically reassortment. So this genetic drift, because basically what was happening, this pig was being literally infected with H3N2 from humans, and this H1N1 virus inside the swine. So you have 3 different viruses, and these 3 viruses came up with a new H1N1. This was a product of RNA reassortment, so antigenic drift. Shift. So this really caused a new H1N1 virus that was adapted to actually kind of go into humans. So this is again a swine influenza that was not able to bind to humans. Now it is, and it was causing very similar symptoms to avian influenza. It wasn't as bad as your H5N1, but still was a huge, basically kind of, let's say, it did affect the world to a degree. So this is again what we're trying to say in some sort of way, where basically here in this case of the basically this H1N1, this pig literally got these viruses from the H1N1 and basically also from the human that literally caused this huge reassortment. So you created a new basically virus from the pigs that was able to jump into the humans. So basically this is the end when it comes to influenza viruses. Now we're going to talk about more paramyxoviruses. So paramyxoviruses are very similar per se towards your RNA viruses, to your influenza. They are enveloped, they are single-stranded sense, they're single-stranded RNA viruses and negative sense. So again, very, very, very similar towards your basically influenza viruses. But basically what's something quite cool from these paramyxoviruses that's very, let's say, kind of characteristic is they're able to do synthetia. So remember, they're able to create multinucleated cells. They're able to make cells fuse together. And this is extremely important hallmark for paramyxoviruses. Specifically, can help us a lot with when it comes to actually diagnosing. So again, paramyxoviruses, they basically do a very similar to influenza viruses. They will actually bind towards, in some sort of way, to the host cell through using their glycoproteins. Rather than endocytosis, they will literally kind of fuse with their membranes and release RNA. So their RNA, per se, now this time it doesn't go to the nucleus and will basically just do its transcription and translation within, let's say, your host cell. The issue with parainfluenza viruses, basically, it usually kind of stays mostly up in the upper respiratory tract. It usually is a major cause of respiratory infections, specifically in infants and young children, and it has different clinical symptoms. Basically, parainfluenza is known to actually cause croup. What is croup? It's also known as this laryngotracheobronchitis. So basically what that means is that you have your larynx, your trachea, and your bronchioles, all of them infected. And what happens with these kids is you literally have this raspy voice. There's basically difficulty of breathing. So they produce a very, let's say, unique type of symptoms. So again, you get these kind of characteristics of group, this barking cough, this hoarseness, this basically inspiratory stridor, and high-pitched sound coming from your airway obstruction. So this is usually what comes from basically this parainfluenza. So parainfluenza, as I said, it usually leads just in your respiratory epithelial cells, mostly up in your upper respiratory tract. So of course, just basically they are mostly kind of removing your epithelial cells, causing cell damage, and removing your your mucus. But unlike influenza, parainfluenza is not severe. It's just very localized and mostly towards your upper respiratory tract. What's mostly very important parainfluenza is the syncytia in general. So in generally, we do have an immune system or immune response. We have antibodies, we have also T-cell responses, but they're very short-lived. And generally, they're not that strong. So what happens is that in generally, after 2 or 3 months, our memory cells tend to basically die. They are not that good at inducing good memory responses. And because of that, we are basically quite constantly infected with parainfluenza in general. Then we have another type of this paramyxovirus, which is RSV. So RSV basically we have definitely spoken about this. It's a very common virus. It usually causes basically a lot of lower respiratory tract infections in infants and in young children. So it can be quite dangerous. By the age of 2, every single baby has had RSV. So this basically it's a enveloped virus. It's single-stranded negative sense RNA virus, super contagious, and can be literally very dangerous when it comes to actually kids, because they can go to the lower respiratory tract. And generally what we have a lot of reinfections because, again, similar to what you see with the parainfluenza, we do not have quite a strong and long-lived immunity. We actually have a very short-lived immunity, so we get infected with RSV quite constantly. So the structure of, or what's unique of RSV is usually they have this fusion protein, this F protein, that is really important when it comes to fusion. So we have the attachment protein, the G. So this is the one that attaches towards the receptors. And then you have the fusion protein. And this fusion protein, really what it does, it helps fuse the membrane of the virus with the host membrane. But of course, this fusion protein, what it can also do is induce fusion of infected cells. So creating literally syncytia, basically syncytia, and helping to create this multinucleated cells. So as I already said, the RSV infection usually mostly happens in the lower respiratory tract. And you have quite a lot of changes there. You actually have quite a lot of cell death, quite a lot of inflammation, and an increased mucus production. And because of these three things, what happens is you have a whole rearrangement in your bronchi, which can cause a lot of issues, specifically when obstructing our airflow and causing difficulty of breathing. So what we see a lot with these kids that are infected is we see bronchiolitis, and we see wheezing and respiratory distress. So it can be quite actually dangerous. In majority of the cases, it usually goes on its own, but in some cases, it can be quite severe and can cause quite a lot of damage within the lungs. So it really can cause a lot. It can cause death. So usually, our immune system that is basically actually, let's say, developed, is not that protective. So it allows for reinfections because, again, the immune cycle is not that good when it comes to inducing a proper immune response. And most of this is coming from the syncytia formation. Because we have syncytia formation, they are very well hidden from your immune response. So this is why we always have a lot of this RSV infections coming up and up and up again. But also RSV is able to kind of evade our immune responses as well. So in generally, prevention is the key when it comes to RSV. So if we know that a baby has some sort of predisposition for mortality because of an RSV infection, like congenital heart failure, or they're underdeveloped, we usually give them pavlizumab. Pavlizumab is basically an antibody against the F protein, the fusion protein, that stops the RSV from actually infecting. But of course, in generally, when these kids are infected and they have not been taking pavlizumab, the treatment usually includes oxygen therapy and hydration because these kids require it. And generally, antiviral therapy for RSV is very limited. You really don't use it. It's mostly making sure that this baby does not die because of a lack of oxygen. Another basically type of paramyxoviruses is your human metapneumoviruses. These are very similar to RSV, but in generally they cause very similar respiratory illnesses, but in generally they are very, how can I put it, not that dangerous. They do not really cause much damage like your RSV. So they can cause damage in elderly or immunocompromised patients or children, but to a lower extent. It's quite low in generally. They can basically kind of go in both in your upper and lower respiratory tract. And generally, as I said, the clinical presentations very similar to RSV. You have your cough, your fever, your wheezing, and difficulty of breathing. But in generally, it's much more less severe than RSV. So notroviruses can cause significant disease, particularly in vulnerable populations like elderly people or children. But again, it's not that severe compared to RSV. So generally, there's no much treatments to do. Reinfections are again very possible because it's incomplete immunity. Because again, these metapneumoviruses can cause this syncytia, so they really can evade your immune system and make your immune system quite low in the production of memory cells. Now we're going to a different kind of family, which is your rhinoviruses. These are the most common viruses. These are belonging to your picornaviruses family. So picornavirus family is quite big, but one of the most important ones from picornaviruses is rhino. These are non-enveloped, single-stranded, positive sense RNA viruses. And because they're unenveloped, they're highly stable. So they're able to really be on literally kind of surfaces for quite a long time because they don't have a lipid membrane. So they're not that, let's say, sensitive towards the environment. The issue with also rhinoviruses, there are about 100 different type of serotypes. So different types of rhinoviruses with different mutations in their basically basically kind of outside proteins. So this is why, in generally, we do not have an immune system that gives us a long-lasting protection. So we get rhinoviruses quite often. And usually when we talk about the common cold, it's rhinoviruses because it's so common to get infected with rhino. But the good thing of rhinoviruses, they're very limited towards your upper respiratory tract because it loves to basically replicate at 33 degrees Celsius. And that's only found in your upper respiratory tract. Your lower ones, it's much more 34, 35, and 37 degrees. Around those areas, it doesn't really like to replicate there. So it really stays up there. So because it stays up there, usually your, symptoms are quite easy. They're, very your common cold symptoms like runny nose, sore throat, and a cough. So you do not get these very kind of nasty symptoms like, that you get from influenza, like high fevers and severe muscle aches, which are usually caused by having a massive immune response in basically our lower respiratory tract. Usually in your upper respiratory tract, you don't get much of a higher immune response over there. Then you have adenoviruses. Adenoviruses are very different. They're non-enveloped, but they are DNA viruses, and they're double-stranded. Because of their non-enveloped, they're very resistant towards environmental conditions. They make them survive on surfaces for quite a long time. And the way they can transmit is through fomites, and they're also able to through basically, through touch, through towels, medical instruments, and contaminated hands. So they're very infectious. And usually they're able to basically, of course, infect your upper and your lower respiratory tract, but generally they're not that dangerous. So adenoviruses are quite persistent, and they can be basically sometimes in cause quite a lot of outbreaks in military recruits or schools because they're very persistent cells because they're able to really survive in so many different places. So what are the clinical manifestations of adenoviruses? Again, they're respiratory viruses that cause mostly pharyngitis, bronchitis, and pneumonia. So you really do get a lot of this kind of sore throat and fever coming from that. But the thing about adenoviruses that is a bit weird is that this virus, other than just infecting your respiratory tract, it can also bind to many different types of cells. So it can be systemic. It can go to our stomach to cause gastroenteritis, colitis, and hemorrhagic cystitis, so more in the bladder. So because the adenovirus is able to replicate not just in our respiratory tracts, it's able to go to different organs and do quite a lot of binding, basically killing over there. So adenoviruses, as we said, it's mostly, infects epithelial cells and induces a lot of the cell lysis. So it causes a lot of tissue damage and inflammation. And it's these two that are causing a lot of the disease per se. So again, the virus is non-enveloped, so very stable in harsh environments per se. So in generally, immune response is really good at controlling the infection, but again, the virus has some mechanisms of evading the host defense. So generally, adenovirus produces extremely good immune responses, but because the adenovirus is able to also evade the immune system, it's able to keep persisting. But generally, adenoviruses are very strong inducers of our immune system. So in generally, our diagnosis with adenoviruses is through PCR. Usually that's in generally how to do that. Generally, there's no widely vaccine for adenoviruses. There is an oral vaccine for military personnel to prevent outbreaks, but in generally, you do not really use them that much. So now going into coronaviruses, which I'm quite sure quite a lot of you guys are quite interested due to the pandemic that we basically recently had to go through. So coronaviruses are enveloped, single-stranded, positive sense RNA viruses. And these are characterized when you look at them through a nice microscope. So this is basically your surface electron microscope. You get this really nice formation of these beautiful crown-like structures. Hence, corona. Corona means crown in Spanish. So you have these beautiful crown-like structures that hence gives it its name. And these are the structures, these basically spikes, we call them these kind of spike glycoproteins, are the ones responsible for binding to the host cell receptor. And the coronaviruses, as we know, they're very important viruses that bind to respiratory, but they can also go down to our gastrointestinal system. So I don't know if you guys remember, there was one of the coronaviruses that was able to induce gastrointestinal symptoms like diarrhea or upset stomachs. I think, if not mistaken, it was the Delta coronaviruses. So usually their tropism is mostly towards your respiratory tract, but they can go inside your stomach. And the symptoms can come from very mild cold symptoms to high severe respiratory diseases that we know of. So, in generally, you have different types of families. These basically alpha, beta, delta, basically, or basically this families that you have. But what we see is these alpha COVID viruses, specifically this 229E and OC43. These are coronaviruses that usually are very mild. They only basically infect your upper respiratory tract, and they very gonna give you very similar common cold symptoms. Again, they are very short-lived immunity because most probably they're just very limited towards one area and your immune system is not that activated that much, and they're basically removed. So because we don't have a strong immune system response, we tend to be reinfected constantly with these coronaviruses because our immunity is not that active or that strong or long-lived. Then you have the severe, which we know as the SARS, the MERS, the COVID-19. These guys really are able to go to our lower respiratory tract, and they're able to cause pneumonia and acute respiratory distress syndromes. So this is why, for example, with COVID-19, we started to have a lot of death because they were able to go to your lower respiratory viruses to cause pneumonia and basically literally prevent you from breathing. So coronaviruses, usually, as we know, they basically bind to specific host receptors. For example, we know that SARS-CoV-2 uses the ACE2 receptor to enter cells, and once inside there, it literally replicates in cytoplasm and can cause direct cytopathic effect. So itself can destroy basically your host cell, but it can also induce your immune system to cause damage. So it's both the virus able to directly kill your host cell, but also induce your immune system to cause quite a lot of systemic damage by releasing a lot of cytokines per se. So the transmission of basically viruses, as we know, these coronaviruses are through respiratory droplets. So because of respiratory droplets, this is why we had to basically move 2 meters away from each other, always. Surface transmission could occur, but it's basically less common. It's mostly, again, through droplets that we did. In general, when we basically, again, these mild coronaviruses like the OC43 and 229E, these ones were very generally easy to be removed. So in general, we do not have a strong, long-lasting immunity. So we always get an infection with them. But the SARS and basically MERS and basically the SARS-1 and SARS-CoV-2 viruses, because they produce such a high immune response, we can actually get a very long-lived memory response. And this is why we then developed actually vaccines, because we are able to induce a very strong immune response. But the mild ones we're not able to, so creating a vaccine is useless. So that's why we have mostly vaccines go towards the very, let's say, pathogenic coronaviruses. So guys, thank you so much for listening. And if you have any questions, please do let me know.